Give me a minute here. Can somebody confirm you can hear me?
I can hear you.
Thank you. Let's see, where are we? There we go. You should be seeing my screen now. Today is February 27th, only one more day in this month, I think. Make sure this isn't a leap year. Yeah, just one more day in February. And we are supposed to be on chapter 14. And we're supposed to finish chapter 14. I think we will succeed in doing that. There is no lab tonight. You should be working on your unknown project if you don't have that project done. Let me remind you, your unknown notes pages will close March 7th, and then the unknown project is due March 8th. Sorry, I'm having a hard time reading that. Um, so that's about a week more on the unknown project. Let me inform a number of you that you need to finish your unknown notes pages. When I sent out the last results, you do need to put in the last results, and you should give your conclusions at that point as well. All right, any questions about any of that? All right, let's begin with chapter 14. Okay, so I have that we had talked about where the human microbiota can be found. Oops, went too far. And then places where they're not found, internal places to the body, like the blood, muscles, the nervous tissue. There are some interesting facts about the normal microbiota and its relationship with its host. First of all, humans have about 3 times 10 to the 13 human body cells. But on humans and in humans, we have about 4 times 10 to the 13th bacterial cells, meaning we have more bacteria cells in and on us than we have our own human cells. And that begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much. Here are some normal microbiota cells. Let me blow this up so you can see it better. Bacteria found in the nasal epithelia. These are the villi of the nasal epithelia, meaning that's part of the human cell. This human cell is actually down here, and then the villi comes up from the cell. But these right here are cocci that are found in the normal um, microbiota of the nasal passageways. That, you don't want to know what that is. Here's some bacteria on the stomach lining. That's the stomach lining there. These right here, that's not bacteria, that's actually villi in the stomach. And you can't make out the cells of the stomach lining, but they're huge. Um, that's probably a divider between one cell and another, but this is a much larger cell than the bacteria. I don't know what that bacteria is. It's obviously a spirochete. And then here we have a normal microbiota in the intestines. And this is close to a rod. That is close to a cocci. And then these are Coccobacillus. This right there is probably food. And maybe there is a normal epithelial cell. It's hard to tell. We found some unusual places for the human microbiota to be. And these are fairly recent findings found around 2014, so a little more than 10 years ago, where living bacteria are found in the bladder of females. And I've looked extensively and I never could find research to find out do human males have bacteria in their bladder. And nobody's ever done the research or at least reported about it. So I couldn't find anything on the question, are there bacteria in the bladder of a male? But at least in females, they do have normal microbiota inside the bladder. And this is part of our normal human microbiota. Females have a shorter urethra, so it is possible that females have bacteria in the bladder and males don't. That is a possibility. And then the other possibility is simply nobody's looked in males, and so that's why we don't know, or at least I can't find out. It was thought that urine was stale when it was in the bladder, but obviously, at least female urine is not. Now, this bacteria in the bladder does not cause a UTI. This is the normal microbiota, at least of females. Any question about any of that? Most of the bacteria are lactobacillus, but there's many different bacteria that are in the female bladder. Let me move to the next slide. They also found bacteria inside mother's milk, even when they sample mother's milk while it is inside the mammary glands of a female. So milk is not sterile, never was sterile, and the mother purposely gives the nursing baby bacteria. It was always assumed that a baby nursing got the bacteria from the outside of the nipple, but in reality, the baby gets bacteria when the baby consumes mother's milk. Any questions about any of that? The number one species was actually bacillus in the breast tissue of a mother, very low, 11.4%, meaning there's a whole bunch of other bacteria that are found there. But all of this bacteria is part of the mother's normal microbiota. And if you don't know, a baby getting mother's milk is going to be protected for two reasons. First, the baby will be getting normal microbiota from the mother, and that's a good thing because the normal microbiota does not cause the baby disease, and it can help defend the baby from other bacteria. I don't remember if we talked about that or not, But if there's normal microbiota around, it can help prevent a pathogen from getting established in the baby's stomach. And then the other advantage of a baby to get mother's breast milk is that the breast milk has antibodies in it. And those antibodies are extremely important in helping the baby defend themselves. And it's been well documented that babies that do not get mother's milk, meaning they get infant formula made by the parents, that these babies are much more likely to get gastrointestinal diseases than a baby that is getting the antibodies from mother's milk. Any question about any of that? So I guess the advantage there is if you can't give your baby mother's breast milk, you should actually spend the money to buy uh, mother's breast milk so that the baby can get antibodies from the mother or from another mother, I guess, from another human female. Any questions about any of that? All right. And the third place that they found the normal microbiota, and this surprises me that they didn't think this would be there, and that is the placenta harbors a unique microbiome. This microbiome is only found on the placenta. And obviously, this is only present, this placenta, when the mother is pregnant. And then when she's not pregnant, obviously, this normal microbiota disappears. Why this surprises me is that the female reproductive system is open to the outside. So it's surprising to me that they didn't realize that bacteria could come up to the placenta. And if you don't know, females' ovaries can get a bacterial infection starting from the vagina, and the bacteria moves its way up. And that's because the female reproductive system is open to the outside. You do not usually want bacteria growing on the ovaries, but some sexually transmitted diseases can get up to the ovaries. Anyways, the point being is that there is normal microbiota on the placenta, and this is part of the normal microbiota, and it's actually healthful for the placenta and then the infant, which is inside the placenta. Is that right? Inside the sac, whatever that's called, which is the placenta. Any question about any of that? All right. So the relationship between the normal microbiota and the host, I guess this is where I'm going to talk to you that now the microbiota is very important for defending the host. For one thing, the microbiota can engage in microbial antagonism with a human pathogen. Microbial antagonism is where two species antagonize each other, meaning they're competing for growing in the host. And so the microbiota can compete with the human pathogen and prevent it from getting established. The normal microbiota can occupy the niche that the pathogen might try to occupy. And if it's occupying the niche, then the pathogen cannot get established on the host. Think of it as a bacteria falling onto my skin. I've got lots of bacteria on my skin. So the chance of the bacteria falling on another bacteria or right next to another bacteria will be very high. And if that were to happen, the pathogen may not get established then. There can be competing for resources. For example, suppose a bacteria falls on my skin right next to another normal microbiota on my skin. The normal microbiota will compete for the resources with the human pathogen. If the human pathogen doesn't get enough resources, it will not get established, will not grow, will not cause disease. The human microbiota can produce acid, making the environment less friendly for the human pathogen. If it's too acidic, the human pathogen cannot grow. Some examples of an acid environment is where the normal microbiota in the mouth create an acid environment. And then also in the urethra, the normal microbiota make the urethra more acidic. And so if a human pathogen gets, I don't know, established, it may be too acidic for the human pathogen to grow and then go on and cause disease. And then further, some normal human microbiota can produce a bacteriocin. A bacteriocin is a protein that prevents the growth of similar bacteria. Like E. coli, producing a bacteriocin will inhibit, the bacteriocin itself will inhibit Salmonella and Shigella from growing near this E. coli producing the bacteriocin. But the bacteriocin will not work on another unrelated bacteria like Enterococcus faecalis, a bacteria found very commonly in the gut. E. coli is also found in the gut. So this bacteriocin created by E. coli is actually a good thing because the bacteriocin will prevent the growth of Salmonella and Shigella, but will not prevent the growth of other bacteria in the gut. Any question about that? Now, if a patient comes down with a Salmonella or Shigella infection in their intestine, they're likely to be given antibiotics. And what are the antibiotics going to do? They're going to prevent the growth of Salmonella and Shigella. They'll also prevent the growth of E. coli, and they'll prevent the growth of any bacteria in the gut, assuming that it's a broad-acting antibiotic, meaning they could use a neuroacting antibiotic that only hits gram negatives, but that would still hit E. coli, Salmonella, and Shigella. If they were instead to use a bacteriocin, it would only hurt Salmonella and Shigella and a related bacteria. It would not hurt like Enterococcus faecalis, which is, I believe, the number one bacteria you can find in fresh feces. It may be the number one in the intestine. I've often wondered why they don't use bacteriocins when a patient gets an infection. And I have read, oops, come on. I have read that bacteriocins are now being used experimentally for infections. So they are being used experimentally to control infections. And they're very good because they work like an antibiotic, but they're much, much narrower in what they work on. So instead of giving a antibiotic works on gram negatives, if we were to give a bacteriocin produced by E. coli, it would only work on Salmonella and Shigella. Any question about any of that? All right. Probiotics is the consumption of live microbes which are intended to exert a beneficial effect. For example, people who eat yogurt having live microbes or a probiotic in it will get the microbes colonizing their gut and then will spread beyond the gut, including growing in the female reproductive system, or at least part of it, and that would reduce the chance of a pathogen getting established in, well, both the gut and the female reproductive system. I think it also reduces the risk of getting oral pathogen as well. Like a yeast infection is much less likely to happen in a woman who is eating yogurt with live cultures. So that's one of the advantage of eating probiotics, or in this case, yogurt. Any question about that? All right. The relationship between the normal microbiota and the host is one of symbiosis, where two different organisms or populations live together. So the microbiota lives on the host and the host lives with the microbiota, so they're living in symbiosis. This relationship can be further characterized as either a mutualistic symbiosis or commensalistic symbiosis. Mutualistic symbiosis, also called mutualism, is where both organisms benefit. Some of the normal microbiota benefit the host, and then the host benefits the normal microbiota because the host provides a home for the normal microbiota and usually provides food. So in the case of mutualism, the bacteria is getting benefit from the host, getting a home and food, and then the mutualistic bacteria benefit the host. One way this is done is vitamin K. And I used to know the other vitamin. What the heck is it? One of the B vitamins. It's eluding me what it is. It's thiamine or niacin or riboflavin. One of the B vitamins. We actually get vitamin K and one of the B vitamins from the bacteria living in our gut. And it's true we don't get enough to get 100% the recommended daily dose, but it's a substantial amount of the vitamin K that we get and the B vitamin as well. It's something like about a third of our recommended daily dose. So that does benefit the host. There are other ways that the normal microbiota can benefit the host, and I've sort of outlined them here. Competing for resources, occupying a niche that the pathogen might have, producing acids, making the environment less favorable for the pathogen to get established, and then producing bacteriocins, preventing the growth of certain bacteria such as Salmonella and Shigella. A commensalistic normal microbiota is one that benefits from the host because the host provides a home for the bacteria, and then the host also usually provides food for the commensalistic bacteria. However, the commensalistic bacteria relatively leaves the host unaffected, meaning the host isn't harmed, but the host doesn't get much of a benefit. And I'm saying much of a benefit because if you look really hard, the normal microbiota could always help the host by microbial antagonism. One example of a commensalistic organism is in each of our eyes, we have one nematode, which lives in the eye. The nematode doesn't do any harm to the patient. So the host is not harmed, meaning the worm does not eat any human cells. What I assume the worm is eating, the nematode, is it must be eating bacteria that fall into the eye. So no harm to the host, and then the host, they mean the organism gets the benefit, and that is at the very least, it gets a home to live in. Any question about any of that? And yes, you have one nematode living in your eye. So you have two nematodes, one in each eye, at least on average. If there's more, there won't be enough food for the nematode, and so I guess both of them would die, or at least one of them would die. No idea how the nematode gets there, but we have one nematode in our eye, and it's a commensalistic organism. I don't know of any bacteria that's strictly commensalistic, but supposedly some of the normal microbiota are commensalistic. Oh, here's another benefit, which I didn't mention up here. E. coli in your gut helps you to digest the food, so that benefits the host. That is benefiting the host not by microbial antagonism, but by direct benefiting the host. And then the E. coli, which is a mutualistic organism, gets a benefit from the host, and that is it gets a home and it gets food. A parasitic symbiotic relationship is where one organism benefits the parasite or the pathogen, and then the host is harmed. And that follows a parasitic life stage where one organism benefits and the other organism is harmed. No normal microbiota is a parasite, but all human pathogens are parasitic, at least in symbiosis. Any question about any of that? Now, usually when we're talking about bacteria, if it causes disease, we call it a pathogen. We don't usually call bacteria a parasite. We use that word to describe a eukaryote, which is giving harm to us, meaning it's a eukaryotic pathogen. We call it a parasite. And I'm sorry, that's a little confusing. It's just that we don't use the word parasite to describe bacteria. We call it a pathogen, a bacterial pathogen. All right, let's talk about the normal microbiota in a little bit of more detail. The normal microbiota does not normally cause disease. However, under some circumstances, the normal microbiota can cause disease. In this case, we call it an opportunistic pathogen, such as when a normal microbiota is not held in check by the host immune system, like in an AIDS patient, and then the normal microbiota goes on and causes disease. So usually the normal microbiota is an opportunistic microorganism, mainly when the immune system becomes compromised, either with AIDS or some other compromising of the immune system, like maybe therapy, like chemotherapy often weakens the host immune system. And then the opportunistic microorganism can be a normal microbiota, but it's growing in a location the normal microbiota should not be. Like E. coli is normally found in the gut and is a part of all humans' normal microbiota. However, if E. coli is found outside the gut, such as in the ear or in the lungs, then it can cause disease because it's not supposed to be in the lungs, not supposed to be in the ear. And then Staphylococcus aureus is normally found in about 50% of people in the United States in their nose and their throat. But if it's found on the skin, it can cause disease. It doesn't mean that it will always cause disease. And Staphylococcus aureus is actually found on the skin of about 20% of Americans. And these people are much more likely to get a boil. This is a small skin infection, which is actually caused by Staphylococcus aureus. Any questions about opportunistic microorganisms, which can be normal microbiota, and they became opportunistic pathogens? All right, if there's no question about that, let's change gears and talk a little bit about Koch's postulates. This term, we have talked about Koch's postulate 2 or 3 times, and by the end of the term, we'll talk about Koch's postulate 3 times. I know we talked about it the first time in chapter 1, and I don't know if we've talked about it another time or not. Somebody might be able to tell me. But this is either the second or the third time we will talk about Koch's postulate. Koch's postulates were used to prove the cause of an infectious disease. And Dr. Koch used the postulates to prove that Bacillus anthracis causes the disease anthrax. Let me blow this slide up so I can read it easier. Move that over here. Oop. So in proving Koch's postulate, Dr. Koch first isolated the pathogen, grew it up as a pure culture, and he got this pathogen from a sick creature. So he got it from an animal, I don't remember which animal he got it from that had anthrax. And then he isolated it and grew it as a pure culture. And then he inoculated the bacteria into a healthy individual, or in this case, with anthrax, he injected it, I think, into a mouse. This picture is showing you a bunny. And then he observed that the animal became sick. Why the bunny is shown on its side. Because it's a sick bunny. And then furthermore, Dr. Koch stated that you have to do more than just state that when we gave the bunny the pathogen, the bunny became sick. You have to prove from the sick bunny that it has the pathogen that you put in it before you can say that the organism causes a specific disease. And then all organisms with this disease must show that they contain this pathogen. And that's Koch's postulate. The different steps used to prove a given organism like anthrax causes a given disease like, a given organism like Bacillus anthrax causes a given disease like anthrax. Any question about any of this? Well, there are exceptions to Koch's postulates. For example, what if you have a human-only disease, meaning it only causes disease in humans, only grows in humans? Could you inject the disease into people, then check to see if the people get sick, and then go through Koch's postulates? Like HIV, have we ever injected HIV into a patient to see if they then later get AIDS? To prove that HIV causes AIDS? Have we ever done that? Anyone want to guess?
No.
Yeah, we've never done that for moral and ethical reasons. So what do we do? Well, there are exceptions to Koch's postulates, and that's one, something like HIV. The disease is so horrible, we would never inject it into a patient for moral and ethical reasons. So how do we know that HIV causes AIDS? We went through very large epidemiological studies and then followed these patients over time. Some of the patients who were HIV negative at the start of the study, sometime in the study, became HIV positive on their own. And then we followed those patients over time, and many of them went on to develop AIDS. And then when we check, all of the patients who have AIDS have HIV. the virus. So that's how we can prove that HIV, the virus, causes the disease AIDS, because you can't use Koch's postulates. And what do you do when multiple organisms can cause the same disease signs and symptoms? Koch's postulate states that all individuals having this disease must contain this one organism. Like the human cold, there's more than one virus species that causes the human cold. In fact, there's 3 different virus families that cause the human cold. So there's many different species. So you can't use Koch's postulate to prove that a certain cold virus causes the human colds. And once again, we have various epidemiological studies to show that a cold virus can cause the human cold. And then because the human cold is not very severe symptoms, there are patients who have volunteered in a study and they gave certain cold viruses to these patients. And many of them, not all of them, but some of them did develop the signs of a cold. And so we can prove that when there's multiple organisms causing the same disease, we can show that there are different organisms that cause that disease. And that doesn't use Koch's postulate because Koch's postulate is so strict. And then what is it when there's a given disease which is caused by one organism, but this one organism can cause more than one disease? Like salmonella can cause salmonellosis, and it can cause typhoid fever, depending on how severe the disease is. And so you can't use Koch's postulate to show that salmonella is causing salmonellosis because there's actually 2 diseases that salmonella causes. Any question about any of that? And then there's another exception, which I don't have here. Oh, right there it is. So I do have it. And that is, what do you do if the microbe has a unique culture requirement, such as a microbe that we cannot grow in tissue culture? You can't use Koch's postulates because Koch's postulate assumes you can isolate it, grow it up, and then inject it into a healthy organism. It used to be that syphilis could not be grown in tissue culture. And so, once again, we can't use Koch's postulate, at least we couldn't, to show that syphilis, the bacteria, causes the disease, syphilis. And I've forgotten the name of the bacteria now. Treponema pallidum. Or is it Treponema? No, it's Treponema pallidum causes syphilis. And until recently, we couldn't use Koch's postulate because you can never get it to grow. However, in the last 10 years or less, people have finally figured out what you have to add to get that bacteria to grow in tissue culture. And then another one is Bartonella henselae. We cannot grow it in tissue culture. And it causes human disease. And if you don't know what that is in humans, it's called cat scratch fever. I'm not certain if they have found a way to grow cat scratch fever bacteria or not, Bartonella henselae, but at least 5 years ago, there was no way to get it to grow in tissue culture. All right, any questions about Koch's postulates? You can expect a question on the final on Koch's postulates because, like I said, we've talked about this topic at least 3 times this year. Or will. Let's talk a little bit about classifying infectious disease. A symptom is a change in the body function that is experienced by a person as a result of a disease, but it is a subjective symptom so that it is not apparent to an observer. Any observer cannot know or prove that the patient has a symptom. A classic example is a headache. When a patient has a headache, only they can know they have a headache. Nobody else can measure that headache. A sign, on the other hand, is a change in a body that can be measured as a direct result of disease. For example, when a patient has a temperature, this can be measured by another individual. So we call it a sign, an objective symptom, no, an objective sign of disease that can be measured. So a temperature of 102 degrees is a sign. If you only have a headache, it is a symptom because there's no way for anyone to verify that the patient has a headache. A syndrome is a specific group of signs and symptoms that are characteristic of a disease, such as the signs and symptoms characteristic of AIDS. A communicable disease is any disease that can spread from one host to another. Measles, TB, and chickenpox are all examples of communicable diseases. A contagious disease is a disease that is easily spread from one patient to another. So communicable and contagious are similar, but they are different. A contagious disease is always communicable, but not all communicable diseases are contagious. Can anyone name a communicable disease which is not a contagious disease? Anyone have a guess? Well, I'll tell you, it's one of these three right here. Got a 33% chance getting it right. Nobody's going to
TB?
guess. Well, it's not measles because measles is very contagious. It's not chickenpox because it's very contagious. It's TB. Tuberculosis is communicable, but it is not easily spread from one patient to another, so it is not contagious. Another example is the flesh-eating disease, which I just had it on the tip of my tongue and then I forgot. Not flesh-eating disease. Well, it is a flesh-eating disease. The disease of biblical times when people would go around saying, I'm unclean, to keep people away from them.
Leprosy.
Is, come on, somebody's got to say it here.
Is it leprosy?
Leprosy, thank you. I just couldn't remember the name of it. Leprosy is a communicable disease, but it is not contagious. You need long-term exposure before you will get the disease leprosy, as well as the fact the majority of the people cannot grow Mycobacterium leprae, so it will not ever grow in the majority of the people. Only a minority of the people can grow the bacteria and then can become a leper. But it is communicable. It can spread from one patient to another. There are also non-communicable diseases. These diseases do not generally spread from one host to another. An example of a non-communicable disease is tetanus. A patient who gets tetanus will not spread that to another patient, except under one rare circumstance. And then a patient who gets botulism will not spread that to another patient, at least normally. There's only one case, one example of a non-communicable disease capable of spreading to another patient. And that is, if the patient were to have a non-communicable disease like tetanus or botulism, and then a cannibal were to eat the patient, the cannibal may get the non-communicable disease. And that's the only example I know of where a non-communicable disease can actually spread. So if you're going to resort to cannibalism, you should make sure that your host is definitely well cooked so you don't pick up any non-communicable diseases. Any question about any of that? That last part was supposed to be a little bit of a joke, so don't be offended by it. All right, let's talk a little bit about the occurrence of disease. To fully understand the scope of disease, we need to know some details about its occurrence. The incidence is the fraction of a population that contracts a disease during a specific time period. So the incidence is only the new cases of the disease. The prevalence is both the new cases and the old cases of a disease. So the prevalence is the fraction of the population having a specific disease at a given period of time. So in 2007, when people looked at AIDS, the incidence, the new cases of the disease, were 56,300 in 2007. But the prevalence of the disease in 2007 was 1,185,000 cases. And that's because some people had AIDS before 2007. Any question about any of that? Some diseases, we use the incidence to discuss the disease. Like, well, I can't think of it, like measles right now. If you don't know, we're having a close to a measles epidemic in the United States, largely in Texas, but it has spread to a few other states. And then some other states are getting the measles too. It's usually reported as the incidence, the number of cases at the time. And I don't remember how many it is, but it's like a couple of hundred, maybe only 100. I'm not sure what it is, but that's the incidence of measles in the United States. The prevalence is used for some other diseases like COVID-19. They were always using the prevalence to talk about how many Americans came down with COVID-19. And that was the new and the old cases together. Any question about any of that? All right. When we're talking about the occurrence of disease, there's a few different terms you need to know. A sporadic disease is a disease that occurs only occasionally in a population. Like typhoid fever, some people say is a sporadic disease. Every once in a while it occurs, but it's not in the US population, at least at high numbers, all the time. You could argue that it's in the United States population at low numbers all the time. And that's a different argument. We're not going to get into that. And that is if the number is very low, is it a sporadic disease or is it endemic? And if it's very low, it doesn't really matter how you divide it. But with typhoid fever, it doesn't happen in the United States very often. And so it's definitely a sporadic disease. But it does happen all the time in the United States, just at a very low number. And so you can say it's an endemic disease as well. I'm not going to use it that way because I'm trying to make a distinction between a sporadic disease and an endemic disease. An endemic disease is a disease that is constantly present in a population. Like in the people of the United States, we always have the common cold, especially in the winter. And then in the tropics, you always have malaria. So malaria is a disease, endemic disease of the tropics. Any question about any of that? Usually when we're talking about an endemic disease, it's happening at fairly high numbers. Because if it was a really rare disease, like I guess you could say in the United States, every once in a while we get cases of malaria. So you could say it's endemic to the United States because every so often we have malaria in the United States. Most of the people getting malaria go to the tropics elsewhere, meaning outside the country, pick up malaria, and then come back to the United States, bringing the malaria to the United States. That's how most of the cases of malaria occur in the United States. But my point is malaria in the United States endemic? Most people would say no. They'd say it's sporadic. And every once in a while, malaria actually spreads in the United States because we do have the mosquito, which can spread malaria. And malaria used to be found in the United States. They got rid of it by simply draining the swamps, reducing the mosquito population. So it used to be found in the United States and it can spread in the United States. And every once in a while, I think it was last year, but it might have been 2 years ago, somebody brought malaria into the United States. And then a mosquito bit them and then started transferring the malaria to other patients that the mosquito bit. It was either last year or the year before. There were like 20 or 30 cases of malaria being spread by mosquitoes in the United States. Fortunately, the winter came, reducing the mosquito population, and then the malaria became extinct once again in the United States. Any question about any of that? All right. An epidemic is a disease acquired by many hosts in an area in a relatively short period of time. An epidemic is when the disease is also significantly greater than it is normally found. Like we don't normally have an epidemic of influenza in the winter, but every few years we do have an epidemic. And what we say is that the cases of the flu are significantly higher than we expect. And that's when we use the term epidemic. And I don't remember the last year the United States had a influenza epidemic. Does anyone know, was there an influenza epidemic recently? Or was the last time before COVID-19 and the pandemic? Let me look that up. Oh, that's not true. That's 2009 was the last big flu pandemic. Let me say in the United States. I'm still saying 2009 was the last big flu epidemic in the United States. Any question about any of that? That's actually a long time ago. We're way past having a flu epidemic. On average, we get a flu epidemic about every 10 years. And that's been close to 16 years from now. What happened probably was the pandemic, not the pandemic, the COVID-19 epidemic reduced the chance of the flu during the COVID-19 pandemic, and so that's probably why we haven't had one um, in at least recent years. Obviously, before the pandemic, it's kind of surprising there wasn't, but it doesn't happen every year. It happens about every 10 years. Where were we? And then a pandemic is a worldwide spread of a disease. So initially, when COVID-19 was first identified in China, it was only in China. It was an epidemic in China. And then it spread to other countries and other continents and became worldwide. And now we call it a pandemic. Usually a disease that is highly contagious, like the influenza, AIDS, even COVID-19. Once they spread beyond one continent, it's called a pandemic. Any question about any of that? All right, let's talk a little bit about the severity or duration of a disease. This is another useful way of defining the scope of a disease in terms of its severity or its duration. We have acute diseases. These diseases, the symptoms develop rapidly. The disease is short-lived. The infective agent quickly climbs in number, causes disease, and then quickly departs from the body, giving a short-term disease. An example of an acute disease is the human cold. You start out with very few viruses, it grows very, very high, and then the virus leaves, and then a short time while the patient no longer has signs and symptoms, and the virus is gone from the patient. That's an acute disease. A chronic disease is a disease that develops slowly and persists for long periods of time. Usually, chronic diseases start very mildly and then over time get worse and worse and worse. The influenza is an acute disease. Mononucleosis, hepatitis B are chronic diseases. We do have diseases in between an acute and a chronic disease. We call it a subacute disease. You don't need to know this term or one of the examples. The symptoms are between acute and chronic in the symptoms. And one example is subacute sclerosing panencephalitis. I'm imagining most of you have never even heard of that. And that's why you don't need to know that. But you do need to know an acute disease and a chronic disease. The extent of host involvement also defines the disease. Infections can be classified according to the extent of which the body's host is affected. There's a local infection where the pathogen is limited to a small area of the body, usually the place where the pathogen got established in the host. A boil or an abscess, that is a local infection, which only involves the skin. Boils and abscesses can be caused by Staphylococcus aureus. And in the case of a boil, it's a local infection. Only one area of the body is affected. However, you can get a local infection become a systemic generalized infection. A systemic infection is an infection that runs throughout the body. Like when a patient gets measles, the measles virus goes essentially everywhere in the body. A focal infection is one where it starts as a local infection, gets worse, and then becomes a systemic infection, usually by invading the blood or the lymphatic system. So if a patient has a boil caused by Staphylococcus aureus, and it gets worse and worse and eventually infects the blood can carry the Staphylococcus elsewhere in the body, and that will be called a focal infection. Any question about any of that? All right. Bacteremia is the presence of bacteria in the blood. This is compared to septicemia, which is actually the growth of bacteria in the blood. Usually when a clinician sees bacteria in the blood, they only call it bacteremia because most clinicians do not have the capability of testing whether the bacteria themselves are growing in the blood. But septicemia is when you can actually show that the bacteria is actually growing in the blood. I guess one way a clinician might be able to know that is if they were to sample the blood, look at it under the microscope, and then they sample the blood later, look at it under the microscope. If there's an increase in number, then they could say septicemia. Usually they only do one, I don't know, assay of the blood, in which case they can find bacteria in it, but they don't know if it's growing. So they usually call it bacteremia. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. All right, the extent of host involvement can determine a disease. The state of the host resistance determines the extent of the infection. A primary infection is an acute infection that causes the initial illness. Like when a patient has Staphylococcus aureus on their skin and then they get a boil. This is an acute or primary infection. A secondary infection is an opportunistic infection after a primary infection has weakened the host, allowing another organism to come in, get established, grow, and then potentially causing further disease. So the primary infection is predisposes the patient getting an opportunistic infection, a second infection, because the host has been weakened. One example of that is where somebody gets a sore throat. Let's say a sore throat from the common cold. The virus weakens the host immune system, which probably doesn't matter, but it weakens the mucus lining. And then another organism can get established, causing nasal infection, nasal epithelial infection, or maybe another throat infection. I'm trying to think of an example of that, and I'm not coming up with one. I can give like smokers. That's not a primary infection, but they weaken their mucous membranes, which then give rise to a secondary infection. A opportunistic organism can then grow with the mucous membranes weakened. That's sort of it. I'm trying to think of an example. Well, the patient gets the cold virus, it can weaken their immune system, and then the patient is more likely to come down with something like strep throat because the number of organisms which can cause disease is reduced because the primary infection has weakened the host, either the mucous membranes or the host immune defense system. A subclinical disease is where a patient has no noticeable signs or symptoms, an inapparent infection, We've talked about this before, but we called it an asymptomatic infection. Remember, I mentioned that young adults, most of you are young, it's estimated that about 50% of patients who are young, when they get infected by COVID-19, they will have a subclinical or subacute disease where they have the infected organism growing in them, but they have no signs or symptoms of disease. And the trouble is they can then spread that disease, which is COVID-19, to somebody else, such as your grandparents, and in which case the COVID-19 would be very severe on your grandparents. Obviously, on young people, COVID-19 in this case is not severe at all because it's not giving the patient any signs or symptoms of disease. And about 50% of young people, both teenagers, maybe older children, not young children, but maybe older children, certainly teenagers and then young adults, will be subclinical for COVID-19. Let's talk a little bit about latent disease. A latent disease is where the patient had the disease carrying organism, and then the disease carrying organism goes latent, not causing any signs or symptoms of disease. A classic example is chickenpox, where the child usually gets the virus causing chickenpox, and then the virus goes latent, usually for several decades, but it could be much less. And then the virus comes out of latency, activates, and then causes the patient to have disease, which will not be chickenpox. Every time the patient gets the disease next after chickenpox, it'll be shingles, where the virus grows down the nerve where the virus was hiding out, meaning it usually hides out in the ganglia nerves, and then the virus can come down the neurons coming from that ganglia. And that's why shingles is usually only in one area, which is innervated by the nerves all from that one ganglia. Anyways, that's a good example of a latent disease. Another good example is the cold sore. A patient has the cold sore from the virus, and then the patient gets better. They no longer have any signs or symptoms of disease. They actually do have the virus, but the virus has gone latent, does not cause disease, is not really reproducing, very low metabolism, if any metabolism. The virus is just hiding out, waiting to come out again. And then the virus activates. That means it leaves latency, goes into its reproductive stage, and then causes another cold sore. There is usually a stressor to activate the latent virus. With the cold sore, the stressor is usually the patient comes down with a human cold, and that triggers the virus to leave latency and then become active. With chickenpox, the stressor is usually simply the patient's old age and their weakened immune system. And then the virus comes out, finds that the immune system can't resist it, and then goes on to cause shingles. Any question about latent disease? If not, let's talk about herd immunity. Herd immunity is when many people in a population are immune to the disease, and that can prevent the non-immune people from getting disease. Let me blow this up so you can see it better. Yep. Trying to move that. There we go. So here we have a population with the immunized people being in yellow, the red people being contagious with the disease-carrying organism. The immune people can protect the non-immunized people, the blue people, so that later on, only people who are non-immunized and in close contact with the infected person will get the disease. Another individual, like this individual here, is protected because for the disease to spread from these people, you have to go to these two, and these two are immune, so they're not going to get the disease. And then the disease will not ever spread to this individual because this individual isn't in close contact with someone who is infected. If you have enough people in the population who are immunized, you create herd immunity, protecting the non-immunized people in the population. The trouble is, and that is actually the point of the vaccine, get the population high enough immunity, and then you prevent the disease from spreading in the population, at least spreading to anyone who's not in close contact with someone who is infected. Because if you're not immunized and you're in close contact with someone who's infected, you will get the disease, even if there is herd immunity. The trouble with measles is you need 90% of the population vaccinated, or measles can spread. 90% is very high, and obviously, measles is spreading in the United States at the moment because there are enough children who are not immunized, meaning over 10% of the children are not immunized to the measles, and then the measles can spread. Any question about any of that? Measles is a very contagious disease, and so you need very high immunity for herd immunity to be in place. What would be a disease which isn't so high? Well, tuberculosis. There is a vaccine for tuberculosis. It isn't used much in the United States, but it is actually commonly used in Europe. And tuberculosis isn't spreading much in Europe because enough of the population is immunized. All right, let's talk a little bit about the patterns of disease. Normally, disease follows a specific sequence of events associated with the infection by a microbe and then the subsequent development of disease. There has to be a reservoir for the disease organism to come out of. The pathogen must be transmitted to a susceptible host. The pathogen then invades the host, multiplies in the host, and then further, the pathogen must injure the host. If there is no injury, the host will not have any signs or symptoms of disease. The illness will then end in one of two ways. The host immune system can destroy the pathogen, so the pathogen is gone, or the pathogen can kill the host, and that will end the illness. Any question about any of that? There are predisposing factors that make the body more susceptible to and the occurrence of disease more likely. For example, females have a shorter urethra than males, so females have a higher incidence of a urinary tract infection. There are inherited traits such as sickle cell anemia, which make the patient more likely to get disease. Another example is diabetics. It's well known that they are more likely to come down with disease than a non-diabetic patient. It's just the disease makes the patient more likely to come down with another disease. Climate and weather. It's well known that colds and flus are more likely to spread in the wintertime. And that's because the climate and the weather. It used to be thought that simply because people were staying indoors, that when the cold came around, People would be indoors and the virus would be more likely to spread. People would have their windows shut. So if there's a virus in the home, it's more likely to stay there and spread to someone who comes into the home. But I don't know, the last 20 years, we realized it's even worse than that. The climate and weather make it more likely for you to get colds and flus in the wintertime than in the summer because one, UV light is stronger in the summer than the winter, and UV light can kill virus. And then further, it's been found, at least with the flu virus, that the flu virus is stabilized by the cold weather. So think of it when somebody sneezes on their hand and then they touch their jacket and then they go outside. The flu virus is more likely to survive in the cold weather. Whereas in the summer, if somebody were to sneeze on their hand and then touch their shirt, because obviously it's the summer, they're not going to wear a jacket, the warmth will quickly degrade the virus, not degrade the virus. The virus just will have a shorter half-life in the warm weather than it will have in the wintertime. And so a patient who's got the cold, they touch their jacket, and then somebody else touches the jacket. They pick up the virus because it's been stabilized in the cold weather. Whereas if someone were to take off the shirt and somebody else were to handle the shirt, well, the virus is likely to be dead because the warmer weather means that the virus has a shorter half-life. So the weather definitely affects whether a person will be getting a disease or not. Fatigue also is a predisposing factor. When you're fatigued, your immune system will be weaker, meaning you will be more likely to come down with the disease. This is often a problem for students because they're often fatigued, not getting enough sleep. I'll talk about that later. Age is another predisposing factor, and that is usually when disease hits a population. Let me see if I can show this. This up. We normally have the incidence of disease something like this. So most diseases follow this pattern where they're very young, and this would be age down here. Let me see if I can get that down. So age on the x-axis. And then this would be percentage infected. Is that the right term? Well, I say incidence. So the very young are predisposed to getting disease, and the elderly. I think I might have shown you this before, and that is we do have the very young for COVID-19, which are, got the wrong one here. Let's do it over here. The very young are, I still haven't got that switched. More likely to get disease, but then the young, as well as teenagers, young adults, are unlikely to have serious disease with COVID-19. And then the elderly are at the greatest risk for COVID-19. And that's because it's very severe in the elderly. Most diseases follow this type of a pattern where the young and the older children, teenagers, are unlikely to get serious disease. But COVID-19, only the very young are at greater risk. The young teenagers, young adults, very low risk of getting COVID-19, serious disease. and then the elderly get very serious disease. There's a few diseases that follow this pattern. COVID-19 is one. Most diseases follow the above pattern. Come on. Lifestyle can affect your predisposing factor. It's well known that smokers are more likely to get a, what do you call it, upper respiratory disease. And that's because the smoking weakens their mucous membranes, making them more likely to get infected by an opportunistic organism. Lifestyle can affect it in other ways too. People who are always partying, meaning they're going out and seeing large numbers of people, they're more likely to come down with a communicable or contagious disease than people who are homebodies, people who stay home, don't have a lot of contact with other people. Chemotherapy also can predispose you to getting disease. It's well known that patients who are receiving treatment for cancer, that many of these anti-cancer drugs weaken the immune system. And so these patients are more likely to come down with a disease. Any question about these factors which can predispose you to coming down with a disease? All right. If there's no questions, I'm going to end it here because I'm a little bit over. I didn't notice that until now. And we will continue with this lesson next time. Any questions before I go?
Have a good night. Thank you.
All right. Bye.